Good afternoon, everyone, and welcome to the divisional briefing for non-clinical development. My name is Brenda Fredrickson, and I will be moderating the session today. So over the last year, the Division of Non-Clinical Development, or DNCD, has been reorganized into three branches. These are the Data Operations Branch, which is led by Jim Little, the Project Management Branch. Thank you. Is this better? Yes. Okay. So we've been reorganized into three branches. They are the Data Operations Branch, which is led by Jim Little, the Project Management Branch, which is led by Dana Harold, and the Non-Clinical Programs, which I lead. So while we've been reorganized, our mission remains the same, and that is to support the non-clinical research across all of BARDA. And because of the mission space that the threat agents within BARDA's mission space, many of them of the medical countermeasures that we support are likely to pursue licensure through the animal pathway. And while non-clinical studies are absolutely important to the licensing of any medical countermeasure, regardless of the pathway, they play an essential role for licensing through the animal route pathway. And that's because in these cases, we need to use these non-clinical studies to enhance our understanding of the pathology of the disease, as well as the mechanism of action of the medical countermeasure. What's more is because clinical trials are often not feasible or not ethical in humans, we need to use these non-clinical models to predict the efficacy of the medical countermeasure in humans. These studies are often very complicated, and one of the things that makes them even more complicated is the fact that they require
The DNCD's approach to providing the non-clinical needs for BARDA has been to develop assays, reagents, and product-agnostic models that we can share with our industry partners to facilitate the development.
And we do this in coordination with other U.S. government agencies to ensure that we minimize redundancies between the agencies, between our efforts, and we maximize efficiencies. And what you'll hear today are examples of how BARDA has collaborated with our USG partners to develop programs to meet the needs, non-clinical needs for SARS-CoV-2, Zika virus, and filoviruses. You'll also hear about the systems and processes that BARDA has put into place to ensure that the integrity and quality of all of the data generated from our non-clinical research, to make sure that the data is fit for purpose. So DNCD's strategy for developing our program has based on 5 pillars. The first of which is our people. So they are the most important component of our strategy. And over the last couple years, DNCD has grown to make sure that we have the appropriate expertise, both technical as well as project management expertise, to meet all of BARDA's non-clinical development needs. We have also expanded our laboratory capabilities to ensure that we have the capabilities and resources necessary for BARDA programs to meet their missions. DNCD continues to develop tools and to adopt innovative approaches, both in research as well as contract management, to make sure that we can meet all of the DNCD needs. And lastly, but not least, is engagement. DNCD engages both with BARDA programs as well as with our network partners and USG partners to ensure that we understand and meet all the needs for clinical development for medical countermeasures. And so with that, I will turn the microphone over to, Katina Wright, who's going to tell us about our clinical laboratory networks.
Thank you. Good afternoon. I'm Katina Robinson Wright, and I serve as the Contracting Officer's Representative for the Non-Clinical Development Networks. Did the slide go? There we go. In 2011, BARDA sought to establish a non-clinical center of excellence in addressing challenges in medical countermeasure development. These networks promote development and establishment of drug development tools, including data, models, assays, and reagents for the medical countermeasure advancement and evaluation. Since their inception, BARDA has invested over $200 million and awarded more than 100 task orders to network partners. The data emanating from these partnerships have supported approval and licensure decisions and have provided key proof of concept data informing funding decisions for countermeasure programs. BARDA seeks to leverage the combined power of in-house, interagency, and industry expertise to assist the development and production of priority countermeasures in a manner that is timely, reliable, and cost-effective. These services are intended to build efficiencies into the enterprise and ensure BARDA has access to critical capabilities as it prepares and responds to CBRN, pandemic influenza, and emerging infectious disease threats. BARDA currently maintains a key non-clinical partnerships with industry and academic contract research laboratories, either by prime or subcontracting agreements. These partnerships represent both domestic and international laboratories and include facilities with high biological containment capacity and the capacity to address chemical agent and radiation and nuclear threats. This fall, BARDA awarded 3 new non-clinical development networks: biological, chemical, radiological, and nuclear. This slide identifies the partners in the non-clinical development chemical network: TNO, MRI Global, Battelle, and Lovelace Biomedical Research Institute. Here we have the partners that are in the Radiological and Nuclear Network: University of Maryland in Baltimore, SRI International, University of Illinois Chicago, and Lovelace Biomedical Research Institute. Here, the partners that are in the Biological Network: MRI Global, Battelle, Southern Research, Texas Biomedical Research Institute, UK Health Security Agency, and Lovelace Biomedical Research Institute. Excuse me. The non-clinical development networks are an internal non-clinical resource. By providing this direct support to BARDA programs, the division aids in the advancement of BARDA medical countermeasure development projects and has helped develop and implement a non-clinical response readiness strategy to address our priority threats. I'll turn it over now to Ashley.
Hello, I'm Ashley Smith in the Division of Non-Clinical Development. In response to the SARS-CoV-2 pandemic, BARDA worked with our network and USG partners to develop the assay and reagents necessary to establish 2 animal models of infection, including a non-human primate and hamster model. The rhesus macaque model shows very mild or minimal disease signs with robust viral replication from the upper respiratory tract via the collection of nasal swabs, and from the lower respiratory tract via collection of bronchoalveolar lavage fluid. The ability to detect viral replication signals and availability of immune reagents makes this model suitable for evaluating vaccine candidates to SARS-CoV-2. Based on this data, 4 of the USG's vaccine partners agreed to use this model to assess the immune correlates of protection for their lead vaccine candidates. This study was conducted following GLP, and samples were analyzed using validated assays developed by the Division of Non-Clinical Development in collaboration with NIAID to ensure data quality and integrity. We also took the opportunity to conduct system serology and cellular immunity analysis in order to gain a broader understanding of the immune response to these vaccines. Our partners will present the results of this study at a breakout session at 1 p.m. tomorrow. The golden Syrian hamster model of SARS-CoV-2 infection shows increased respiratory disease signs with viral replication in the lung and nasal turbinate and weight loss. Based on these parameters, hamsters are a reasonable model for screening candidate therapeutics, as well as testing novel vaccine platform approaches and alternative routes of administration. The Division of Non-Clinical Development took advantage of this model to compare the pathogenesis of the variants of concern listed here on the right, using fit-for-purpose assays developed within our network, and we expect to complete the study analysis within the next couple of months. BARDA's Division of Non-Clinical Development has also supported medical countermeasure evaluation for Zika. During the Zika outbreak, BARDA collaborated with NIAID to develop a program to generate reagents, assays, animal models, and a preclinical data package to support approval of Zika vaccines through the accelerated pathway. BARDA used our IDIQ network to produce a well-characterized challenge stock, while NIAID used their IDIQ network to develop, optimize, and qualify virological and immunological assays. These NIAID-supported assays were then used to establish a non-human primate model for Zika, as well as conduct a natural history study. BARDA has also incorporated passive transfer studies into our program to identify immune markers to predict clinical benefit, while NIAID used these reagents, assays, and models to conduct proof-of-concept studies for early-stage vaccine candidates. The intention of this program was to make assays, reagents, and protocols, as well as preclinical data available to all developers. The sporadic nature of filovirus outbreaks continue to make it challenging to license medical countermeasures against filoviruses using the traditional pathway. As such, BARDA has been working to establish well-characterized models of filovirus infection to support licensure of products under the animal rule. Central to these efforts has been the production of well-characterized, low-passage challenge viruses and establishment of an appropriate animal model with optimized and qualified assays for both Sudan and Marburg. These efforts have been carried out by the University of Texas Medical Branch Office of Regulated Non-Clinical Studies, the Texas Biomedical Research Institute, as well as Boston University's National Emerging Infectious Diseases Labs, in an effort to establish well-characterized models at multiple locations. BARDA's also conducted natural history studies for both viruses. These natural history studies are intended to establish appropriate parameters for evaluating vaccine and therapeutic interventions and provide the data necessary to establish predictive benefit of medical countermeasures to filovirus infection. As with other programs, BARDA's intention is to make these reagents, protocols, and data packages available to all product developers. You can also learn more about the challenges associated with developing standardized research tools at tomorrow's breakout session at 3:15 to 4:15 p.m. And finally, in addition to the programs I just described, BARDA's Division of Non-Clinical Development also supports non-clinical studies across BARDA. Shown here is a snapshot of some of the studies that we support. These studies include the characterization of ventilator-associated bacterial infection, long-term stability testing of medical countermeasures, evaluation of monoclonal antibodies, and a lethal aerosol challenge of Bacillus anthracis, as well as evaluating smallpox antivirals and a monkeypox model, and evaluation of dose-sparing strategies for anthrax vaccines. I will now pass it on to Tahar Babas.
All right, thank you. So I will use, is it working? Okay.
There, wait. No, press here.
I did it.
Looks like you got a
Is it working? Yeah, working. Good.
Use this mic. It won't stay.
It won't
It's good.
stand. Use this one.
All right, thank you. So I will be using next couple slides to highlight some additional capabilities that the Division of Non-Clinical Development has to offer. So DNCD also provides support for the development of medical countermeasures against chemical threat. BARDA recognized that responding to chemical exposure presents several challenges, including the unpredictability of a chemical incident and the rapid onset of life-threatening injuries, which limits the window of treatment and often results in unsuccessful interventions. Medical countermeasures development is further complicated by the fact that there are more than 200 chemicals on the current DHS chemicals of interest list of concern. However, injuries caused by chemical agents share symptomatology or toxidrome with commonly seen medical conditions, and allowing BARDA to take the approach of treating chemical-induced injuries without the need of identifying the causal agent. So as shown here in this slide, studies conducted under the previous IDIQ contract focused on the development of small and large animal models of chemical injuries. These include respiratory depression in a guinea pig, which is the #1 cause of death due to opioid overdose, and a mouse and swine models of pulmonary injury due to chlorine gas exposure, and a swine animal model of hypoxia induced by exposure to cyanide. And then there's also a mouse and guinea pig models of sulfur mustard toxicities and a guinea pig model of cholinergic crisis resulting from sarin gas. These animal models are being used to screen drug candidates and conduct proof of concept efficacy studies. And moving forward under this under new IDIQ contract, DNCD will continue to support the refinement of animal model for the assessment of candidate therapeutic, including readily accessible FDA-approved drugs used to treat disease symptoms similar to those caused by chemical injury. Next, oh, sorry. So similar approach is intended for DNCD role in support for the development of medical countermeasure against radiological and nuclear threats. Exposure to high doses of ionizing radiation can manifest a multitude of damage, including cutaneous injury, vascular injury, and organ-specific injuries. The sequelae of injuries can overlap with radiation-induced pancytopenia coinciding with organ failures. BARDA has focused the non-clinical network IDIQ partners on the development of harmonized animal models for inflammation, vascular injury, and hemostatic dysregulation. Moving forward, BARDA will establish a process to regularly verify the reproducibility of these models to ensure effective screening and evaluation of candidate medical countermeasures. BARDA intends to use these well-characterized models to assess the feasibility of repurposing existing therapeutics and evaluate polypharmacy approach to counter systemic injuries due to radiation exposure. And I'll pass now to Sarah.
Good afternoon. As mentioned, my name is Sarah Duncan, and I'm representing the Data Operations Branch within the Division of Non-Clinical Development. As Katina previously mentioned, since 2011, BARDA has invested over $200 million into non-clinical research. Data from these investments have been published, have supported funding decisions across BARDA programs, and have supported FDA approval and licensure decisions for priority medical countermeasures, many of which are currently being deployed in response to filovirus and pox outbreaks. BARDA's non-clinical data operations branch identifies avenues through which we can enhance the data lifecycle to achieve and maintain data quality while expanding the utility of this important asset. As you've heard in this presentation, the Division of Non-Clinical Development maintains a broad portfolio addressing CBRN threats areas of interest, with the intention of providing tools for the evaluation of priority medical countermeasures. Data management is essential to measuring success within BARDA's mission, and so, as depicted in this graphic, non-clinical data operations focuses our efforts on data quality, data standardization, and availability or usability of these data.
We support data quality by establishing and overseeing purpose and risk-driven quality management systems for ongoing programs, performing functions such as data management review and QC, data analysis, and study monitoring and auditing. Next, we focus on expanding the utility of our data by moving towards greater standardization. Specifically, BARDA is seeking to leverage the Standard for the Exchange of Non-Clinical Data, or SEND. Expanded to include animal rule studies, in 2019, SEND is providing a powerful tool in the medical countermeasure toolbox. Curating current and legacy data and providing standardized non-clinical data provides power to our programs. With standardized data sets, we are better able to conduct meta-analyses across studies and test sites to evaluate model performance and countermeasure performance within these models. Additionally, adhering to these data standards enables better and faster regulatory decision-making, as FDA now requires non-clinical data supporting regulatory decisions be in SEND format. With this tool, we at BARDA are well positioned for enhancing the regulatory utility of our non-clinical data, better positioned for deep analysis and learning, and also better situated to collaborate with our interagency partners, especially during pandemic and emerging infectious disease response effort. Thank you, and with that, I'll hand it back over to Brenda.
Can you just go ahead and advance to the next slide? Thank you, Sarah. So to wrap up our session, I'd just like to briefly go over the priorities for fiscal year 2023 for BARDA's non-clinical development areas. So within the chemical program, their priorities will be to continue to conduct natural history studies to identify target pathways of treatment and to screen drug candidates. They will also assess the feasibility of repurposing common drugs for chemical indications. The priorities for the RAD-NUC program are to harmonize their acute radiation syndrome animal models across various laboratories and to make sure that these models are consistent across a period of time, so verification of the performance over a period of time. And they will also assess therapeutics for treating hemopoietic injuries. And finally, the biological program will maintain its response to SARS-CoV-2. It will continue to develop medical countermeasures against Sudan and Marburg virus, and the influenza vaccine section will seek to improve the animal models for testing vaccine candidates. And can you advance to the next slide? So with that, I'd like to thank the DNCD team that makes all this work possible. And this includes not only the people within DNCD, but also you can see that many of the people on the column on the right-hand side of the screen are our partners in the Division of Contracting Management and Acquisition, which are essential for us to do our jobs. And so with that, I'll thank you, and we can take some questions. So we'll start with a question for Katina. So are there other mechanisms for engaging non-clinical development divisions besides the IQ contract?
The Division of Non-Clinical Development's networks are an internal resource, so we connect directly with program. However, those with countermeasures for our priority threats can engage with BARDA through the EasyBAA, the BAA, or coming in through TechWatch. So that's how you engage with us.
Thank you. One other question, which I'll take, is can others gain access to the models and reagents that we develop, that's developed by DNCD? And the short answer is yes. You can get access to these reagents if they're developed by BARDA, but we don't have a formal process for requesting those. So if you do need access to any of these models reagents, feel free to contact us directly, and we will get the process started for you and help you make sure you have access to them. And those are the only questions I see. I don't see any other in the chat. So with that, I think we can close the session. Thank you all for your attendance.